Okay, guys, so let's get started. We have to thank Dr. Tsuji because now we have a laser pointer. Okay. Quick announcement. Office hours today will be canceled and replaced with email me to make an appointment if you need me. Because I double booked by mistake. But just so everyone knows. Okay. Oh wait, so I can do this? This is gonna be awesome. Yes.
I have a question about the quiz.
Yes.
It stated that there were multiple attempts to take the quiz.
Right. So in the directions, I always say, take it once and only once. Your first grade will be recorded. If you have technical difficulties, email me first, which means multiple attempts is set so that when you email me, I don't need to pull out my laptop and reset the whole Blackboard so you can take it again. Because the first time I taught this class, I felt like I spent the whole weekend taking out my laptop and resetting everything. It was awful. So it's automatically set for 3 attempts. It doesn't mean you can take it for 3 attempts. That's why in the instructions it says only once, first one's recorded. There's no time limit. You don't need to complete it in one sitting. You have all weekend to do it. So just leave it open however you, I don't know, people have been doing it for semesters. So figure out how to leave it open for the whole weekend. And they don't even need to worry about taking it again because you can keep on going back and checking. Okay? Does that clarify? Okay, sorry for the confusion. I think, because I hate how it tells you how many attempts you have. That's something Blackboard does. I set it so that it's open for 3 attempts so I can give people individual permissions. But it always confuses students. Yeah. Yeah. Okay. Let's see how this works. Whoa! Okay. Sorry, technology. Okay, so recitation starts this week. So hopefully everyone saw that there's stuff on Blackboard for you to read and be prepared. And recitation is just around the corner. When you come in, I'm going to divide you into groups that you're going to work with all semester. And we're going to probably have to rearrange some tables. Bring your Scantrons. There's always people who forget. I'm sure there are people right now going, oh my God, Scantron. Bring them because there's going to even be a quiz today. So you don't have your Scantron, you can't take a quiz. And then of course, come prepared. Read the stuff before you get in that room. Otherwise, you're wasting your time. Sometimes they give you more than what you need. So you'll notice when you open the folder, there's like a little explanation of stuff. There's the primary paper, there's a review paper, and some videos. All that together is to help you understand what we're going to be doing. Usually you really need to pay attention to the first 2 things that are up, unless I say otherwise. But I'm not always going to tell you that, because I really would like you to at least look through everything, because that's how you can get a good idea of what's going on, and you won't be sitting there lost. Because that's bad. No one wants to feel lost. Yeah, I keep wanting to walk back there. Okay. So last time we were talking about just the main concept of gene expression and how it occurs in response to the environment. For example, in response to some kind of signal, some signaling molecule coming from another cell to tell the cell that something's going on in the area. And then we started talking about how we study gene expression. We talked about the northern blot, which is where we're actually isolating our RNA and detecting it with a probe. And then we talked about real-time PCR. And we ended talking about real-time PCR. People say real-time PCR and qPCR, which stands for quantitative PCR. People use that interchangeably. Just kind of keep that in mind, because sometimes you'll hear me say one or the other. It's all the same. And I got a question at the end of class about, what was it about? Who gave me this question? I wanted to explain it. You. You came up to me after class with a good question, right? You remember my, I wanted to share it with everyone else. Something about how protein, it was an example about viruses and the viral transcript getting out of the nucleus even though it wasn't spliced. And the question was about how was there a protein that could help the viral transcript getting out of the nucleus if we're talking about RNA. And so that made me realize, right, that was the question, right? So that made me realize that the way I explained it left out some information that I just took for granted that you guys understood. And since it's not a virology class, you don't know all this stuff. So I want to re-explain it. So it was a practice problem. It was talking about how we would use a northern blot to identify which viral protein was responsible for exporting unspliced transcripts out of the nucleus. And in order to understand this question well, you need to understand that when you infect a cell with a virus comes in and does things its own way. It's not how the cell functions necessarily. So sometimes viruses bring in proteins that they need to do things right away early in infection. Or sometimes viruses will make some proteins, so they'll do transcription and translation, make some proteins, and some of those proteins will go back into the nucleus and help to usher out some of the RNAs that haven't been translated into protein yet. So I think that timing thing confused one of the students in the class, and I just wanted you guys to understand that there's not necessarily an order, like where first, yes, with one gene, per gene you have transcription translation, but if you're talking about proteins helping things to happen, there are proteins that their whole function is to move RNA around in the cell. And so it's not, those proteins might be translated long before they're needed. Well, they are going to be translated before they're needed. So does that clarify any confusion, maybe? Okay. Okay. Let's see. Okay, so this is where we ended. And we were talking about real-time PCR, and I was talking to you guys about how it's basically PCR where you have some kind of fluorescence attached to your product. So as your PCR product increases in concentration, your fluorescence increases in intensity. And it's all done in these optical plates where there's a detector that goes to a computer, and the computer is constantly measuring how much RNA there is based on the amount of fluorescence. And so this is the kind of data you get from real-time PCR. Each individual curve is one PCR product. So you'll do, let's say you've got 1, 2, 3, 4, 5, 6, 7 DNAs of different concentrations, or RNA, cDNAs, of different concentrations that you've put into your PCR. They're going to amplify exponentially, but it's going to be proportional to the amount that you put in. So if you put in a really small amount of DNA, it's going to amplify here. If you put in a lot more, if you put in a small amount of DNA, it's going to take a really long time to amplify and come up here. If you put in a lot of DNA, it's going to quickly amplify and come up here. And so what you'll do is you kind of draw a line called the threshold right across the middle, and where it crosses your threshold, you can count that as what cycle number your DNA is coming up at, and that tells you something about how much there is. So for example, you would say that this light blue one is coming up at around, let's say your line is right here. It's coming up around cycle 20 or so, and this purple one is coming up around cycle 40. And something that takes 40 cycles to be detected, there was like nothing to start with. It took 40 cycles to even detect it. 40 cycles of replication, right? Something that comes up around cycle 20, there was plenty there, and so it came up quickly. If there was even more template added, it would come up at an even lower cycle number, right? If you put tons and tons of DNA, cDNA in there, it would probably come up, around 10 or 14 or 16, and it would be over here, okay? So does anyone have questions about real-time PCR? And yes?
Why are 3 of them converging over 33%?
Oh, we don't even care about that. So the thing is, what you want to know, what you do is this orange line here, what you do is you bring it up to somewhere that's in the logarithmic phase, and that's where you can compare. Because as you get up into this plateau, you can't really tell anything about it. And so you pull it up and you actually compare it around this logarithmic scale, and you notice the logarithmic phase, and they're very distinct lines there. So that doesn't really mean anything because it's getting saturated up here, and that's why they do start to kind of come together. And the further up you go, if you cut, eventually they'll all start to be clustered up here. Okay? Yes?
How do you ensure that a different fluorescence is used for each PCR template?
So you do not, it's not a different fluorescence, it's actually all the fluorescence. And what happens is each template is going to amplify with different kinetics, right? Some are going to be faster, some are going to be slower based on the amount of DNA going in, and it's going to follow its own curve, right? Does that make sense? So these colors are actually pseudo-colored by the computer. Yeah. So that question was how do you have different fluorescence for each different template, but you actually don't. Because if you have a starting DNA, in this primers, that DNA is going to increase according to a logarithmic scale, and the computer is detecting it that whole time. So you're going to see it. So you're not going to get that confusion. Any questions? So really, don't get bogged down with the details. Just understand that you can quantify your transcript levels through real-time PCR because you can design primers, just like with the Northern, you design a probe that's specific to your gene, and because fluorescence is integrated into your product as it's being made, the computer can detect the intensity. So you're able to follow and come up with a concentration for how many copies of RNA you had. Okay, I'm going to move on. Is everyone comfortable? Are you guys happy you took this class? Or are you wondering why you were like dying to get in? Okay. What kinds of questions can we answer with real-time PCR? Okay, so this is work that we do in my lab. We look at a whole bunch. We look at everything ubiquitin, but here we have a project where we're looking at Kaposi sarcoma herpes virus, and we're looking at all herpes viruses. They have latent phases and lytic phases, and we're looking at how KSH3 transitions from latent infection to lytic replication. And we actually use real-time PCR in some of our research. So there's a protein, a latent protein called VFLIP, and it activates gene expression of all these NF-kappa B-induced genes, two of which are TNF-alpha and ICAM1. Don't worry about what they are, what they do. They're genes. They're induced by latent infection with KSH3. And the function of VFLIP and this gene expression is required for latency. And RTA, which is a different viral gene, which actually induces lytic replication, it turns off VFLIP and it turns off all the gene expression. Well, we were asking whether it does, but I'm going to show you what it does. So our question was, does the lytic inducer RTA turn off VFLIP-induced gene expression? And so we took that data, like I just showed you, that had those curves, and we calculated relative gene expression. I won't tell you how because you'll start crying. And then we put it on a graph that's really easy to read, right? So pcDNA is just an empty vector, so it's a control, and each one is a different gene. So this is TNF-alpha expression, this is ICAM1 expression. For TNF-alpha, we had primers that we designed specifically to the TNF-alpha gene. For ICAM1, we had primers specific to the ICAM1 gene. And we took our cells and we did different things to them, right? In one well of cells, like we had, cell culture, we had a 6-well plate. In each well, we did a different thing. In one well, we just put an empty vector control, so nothing, just cells alone, because we need to know what's normal gene expression when you've done nothing to your cells. Then we put in VFLIP, this viral protein, and we needed to know what does this viral protein do. What do you guys think it does? Is it activating or repressing gene expression? Activating, right? So that's what it's supposed to do, right? So it activated gene expression for both genes. And then we put in RTA alone, because it's a control. And we don't want to see RTA doing anything to TNF alpha or IKIM1 expression in the absence of VFLIP, because our whole thing is it's turning off VFLIP. And it doesn't do too much. There's a little bit of activation, but not that much. Then we put RTA with VFLIP. And what's happening here? If you compare VFLIP, which hugely induces TNF alpha and IKIM expression, and to VFLIP with RTA, is it still activating or is it, what's happening? It's turning it off. So does this prove my point that RTA is doing something to turn off VFLIP-induced gene expression? Right. So this is all coming from a few real-time PCR runs. We got a bunch of those curves. We calculated it. We normalized it because we need to make sure it was equal across all cells. And then we made it relative to the VFLIP control, to this one. That's why that's set at 100%. And we were able to say VFLIP induces RTA, down regulates it, fivefold, fourfold. And yeah, about that. Any questions about this? I think it's not very useful to understand a method unless you understand how you can use it. Okay, are we happy? Do we want to cry yet? Anyone crying? Good. I try not to make you cry until after the first exam. Okay. See, there I am walking around the back. I don't need to. Okay. So now let's talk about microarrays. Who loves microarrays? Yay, we have one here. So microarrays are awesome because of their power, and they suck because of the amount of data they give you. Okay, microarrays, they can be used to determine genome-wide, or at least a large subset of genes, for gene expression. So would I use a microarray to look at the role of RTA on VFLIP-induced gene expression? I guess I could, but it would be kind of like using a cannon to try to catch a mouse, right? Try to kill a mouse. It's a really powerful method, but you're looking at a whole genome. So people always like to say, do a microarray when I ask questions about gene expression. You always need to think about what method is useful for the question that you're trying to answer. So with a microarray, you have a chip, so it's like a little slide that you guys use in micro to do your gram stains. So you've got a chip, and on that chip there's actually a printer that spots little oligos that correspond to each gene in the genome, or you can get a transcription factor microarray, you can get viral genome microarrays, you name it, you can get it. But each little dot corresponds to one gene. And then you do your experiment. So you've got some control situation, and then you've got your experiment. That might be uninfected cells and infected cells, or normal cells and tumor cells, or whatever. Tumor cells treated with drug, tumor cells untreated. And so you have your control and your experiment, and you extract all your RNA, and you label them with different colors. You've got green, which looks kind of yellow here. You've got green and you've got red, and you pull them together and you incubate them with the microarray. And if the RNA is present, it's going to bind to the corresponding gene because of that complementary interaction, right? And if it's present in high abundance, a lot of it's going to bind. If it's present in low abundance, not much of it's going to bind. And don't forget, you've got your green and your red together, so they're going to kind of duke it out, and whichever one the most of it is there, you're going to have that corresponding color. And so what you end up with is something that looks like this with dots of different colors. So a dot with red will mean that it has a lot of the experimental RNA bound to it. A dot with green means it has a lot of the control RNA bound to it, and a dot of yellow means that it has both, because red and green, when they're scanned and overlapped, look kind of yellow. So if the spot is red, that means the gene is expressed higher in your experimental than in your control, right? Because there's a lot of a lot of the red one. And if there's green, it means that your gene is expressed higher in your control. And then yellow is expressed the same. So the way you would think about this is upregulation or downregulation in response to whatever condition you're using. So I'll go into this a little bit more. I put an example here, which I thought was a nice one, right? So what kind of questions can you answer? People who work on cancer love to, everyone loves microarray. I kind of hate the idea of them. That's only because if you think about how much data you get, how do you handle that data? You suddenly need to either be good friends with a computer programmer or become a computer programmer. Because it's not data that you and your brain can handle by itself. So here's some data from a paper, and I have the reference below so you guys can look it up. A paper where they analyzed breast tumors from 117 patients to look for poor prognosis gene signatures. Interesting, huh? That's useful information, isn't it, right? You've got all these tumor samples, you do the microarray, whole genome-wide array, and you get your ups and downs of all your genes in your genome per tumor, per patient, and then you do statistics and you can say, oh my God, these people who died all had really high levels of this gene. These people who survived didn't have a very high level of those genes. That must mean something, right? And then you can go on and validate it in some other study. This is really, really useful. You can find something like 30% of breast cancers have high levels of HER2, which is a receptor expression. You can find all this information, and it's really useful not only in prognosis, but also in deciding what treatments to give to some people, because there are specific treatments for specific types of cancers. What else was I going to say about this? So what kind of genes do you think might be expressed at really high levels in tumors in people who probably are not going to do very well? Just if you had to come up with something. Growth factor receptors, probably. Yep. Apoptosis. Do you think apoptosis genes would be upregulated or downregulated? Downregulated. They probably would be, right? Someone you want, like most cancer drugs, most chemotherapeutic drugs are trying to induce apoptosis, and cancer cells like to turn off those apoptosis genes so they can be resistant to those drugs. Yes.
Genes upregulating angiogenesis.
Genes involved with angiogenesis. Absolutely. What's angiogenesis? Right. Creation of blood vessels, because tumors need blood supply. Right, absolutely. Yeah.
Cyclins and CDKs.
Cyclins and CDKs. What do cyclins and CDKs do? Right, so pushing cells into cell cycle, right? Because cancer cells like to reproduce. Anyone else have any guesses? Those are all really good ones. And if you look, you will see plenty of VEGF, plenty of cyclins, definite downregulation of apoptosis factors. Yep. Anything else? Yeah. So this is all useful information. Sometimes you're just proving what you thought anyways, right? But it's kind of nice to say, well, in these 117 tumors, just like I thought, apoptosis was downregulated. Okay, any questions about this? Okay. So in these figures, so this is actually showing, these are all the tumors on one, so each line across is one tumor, and then the whole genome. So the red, those are things that are upregulated, the green are downregulated. And I think they make black no change. Okay, what else?
I do have a question.
Yes.
You said on the chip, there's, how does the RNA bind to the genome?
Oh, right. Okay, so on the chip, you've got oligos spotted, and remember, you have complementary DNAs interacting. So you're going to have what's spotted on the chip is going to bind to the RNA complementary. So it's very specific. Any other questions? Okay. Here's another assay. Reporter assays allow you to analyze promoter or enhancer activity with a visible readout. What does that mean? What's a promoter or an enhancer? People have answered this a few times. Something, so where is a promoter in relation to the coding region of the gene? In the beginning, it's just before the start site, right? And that's where your RNA polymerase is going to bind, right? So reporter assays, actually what you do is you take your promoter and you're putting it in front of some other gene that makes a color change or creates light or whatever, so that you can look at the activity of the promoter by visualizing some kind of color or light change. And so the one I have here is luciferase. Luciferase, you know, firefly luciferase. Does anyone know what firefly luciferase is? Has anyone seen a firefly before? Okay, good. Has anyone seen that light that they do? Yeah. So that's firefly luciferase. That's responsible for the flushing of a firefly. Did you know that different species, they flush at different intervals, and that's how they know to mate with each other? I think that's cool. I'm not like an ecology. I don't know nature the way I would like to, but I think that's so awesome. When I learned that, I thought that was cool. I think crickets are similar. Am I right? The pitch is different, right? Yeah, they don't light up. The pitch is different. I think whenever I learn about that stuff, I'm like, I wish I had taken some more ecology classes or something, entomology or something in undergrad. But I was so like, I need to study this. This is what I want to do. It must go this way. So you guys should diversify so you feel like you know about lots of things. Anyways. Okay, so what you do, so this is your plasmid. This is your reporter plasmid right here, your vector. And you have this luciferase gene here, and here's your cloning site. And you'll clone in your promoter of interest. Let's say we wanted to look at the example we'll have in a minute is an NF-kappa B response element. NF-kappa B is a signaling pathway involved in gene expression, and the actual signaling pathway results in a transcription factor entering the nucleus, binding to a promoter, activating gene expression. So you would put the DNA element that the NF-kappa B transcription factor binds to, you'd put it right before your luciferase gene. And then you can do whatever you want to test the activity of the promoter, because then you'd be able to look at how much luciferase is being produced. Okay, so what you do is you have your control and your treatment conditions, whatever they would be, and you have your promoter because you know what promoter you're looking at. Here it's going to be the NF-kappa B responsive gene promoter, and then you put that before your luciferase gene, and this is all plasmids that you transfect, so you put them into your cells in your cell culture, and you incubate them, and then you lyse them, and if your promoter is active, your luciferase was transcribed and translated, you've got plenty of luciferase protein in your cells, you lyse them, they're sitting there, and you add in the substrate, and it creates light. And you put it into a machine, a luminometer, and it reads the intensity of your luciferase. So if you, so this is what it would look like, right? So you would have your negative control should have no anything, because negative control is negative. And then you would have your reporter, where you add different amounts of your treatment. Here, we're putting in TNF-alpha, where TNF-alpha is a huge activator of NF-kappa B. And if you put increasing amounts of TNF-alpha, you have increasing amounts of luciferase. These are relative luciferase units. It's a measure of the light, that firefly light that's being produced. And that correlates to the activity of your promoter. So if you could also, let's say you don't like luciferase, you want to save the fireflies, no, I'm just let's say you don't like luciferase for some reason, you're just a hater of luciferase, and you want to do GFP, you can do GFP, anything that changes color that you can see and measure in some way. So here's an example where they did GFP, where you have increasing amounts of GFP over time. They added TNF alpha and they took pictures over time, and you have increasing GFP expression. And that's just that's nothing but a reporter. Who cares about GFP or luciferase? You care about your promoter that you put right before it, because your GFP and your luciferase are acting as a reporter of your promoter activity. Okay, so what kinds of questions could you answer using a reporter assay? Well, you could ask questions about what kind of transcription factors activate gene expression, right? Because you can have your cells and you could knock out different transcription factors and see if gene expression still turns on. You can put in different transcription factors and see if you can activate gene expression. You can look at signaling pathways and see if gene expression turns on by activating different signaling pathways. You can look and see how pathogens affect gene expression by looking at promoter activity in the presence of different pathogens or viral proteins. And so that's the example I'm going to give you here. So this is also stuff that we did in my lab. And this is a reporter assay. And again, we got our numbers and we made it in this nice, easy to read graph. Right, so we transfected our cells with the NF-kappa B reporter. So that again is luciferase gene, and right upstream from it is the NF-kappa B response elements. And we put in our VFLIP or our RTA, because VFLIP is supposed to induce NF-kappa B. And you can see with vector alone, VFLIP, big bar, turning on NF-kappa B, we got lots of luciferase. And when we added an RTA, which was supposed to be shutting things off, was it shutting it off? Kind of. And then here we did a control where we put in TNF alpha. TNF alpha is a known inducer of NF-kappa B, and we got lots and lots of luciferase. So that means that TNF alpha was going in there, activating NF-kappa B. Some kind of magic was happening, which I won't bother you with. And then NF-kappa B transcription factor was going into the nucleus and binding to the promoter and activating transcription translation of luciferase, right? Because we put our promoter right in front of luciferase, and we get lots and lots of luciferase activity. Any questions about this stuff? It's useful to understand this because I like to ask questions on exams where I ask you to design an experiment or interpret results of an experiment or something like that. Don't worry, we'll have lots of examples. Okay. Okay. Let's see. Oh, right. Okay, so we're going to go over the quiz briefly. Actually, I want to try something today. Let's see what we can do. I wanted to say, does anyone have a cell phone? I want to see if we can just try this, because what I usually do is have people hold up letters, and I want to see if we can text. Is everyone on an unlimited text plan? I don't want you to incur charges. If you're worried about incurring charges because of my class, feel free to not participate.
There's
No service?
Yeah, there's not service.
For texting even?
No.
Okay, well, so that's why I only did like one or two of these. So I was hoping we could just pull it and then I can get the information. Okay, so we're, huh? You can't see that? I'm gonna have to work that out. Well, it doesn't matter because we don't have service. Good thing I didn't make a full-on poll. Okay. I know, it was gonna be really fun, wasn't it? Okay, let's go back. So much for technology. Okay, when you want to use technology, where is it? It's nowhere. There's no service. Okay, so let's do this. Okay, so everyone write on a piece of paper A, B, C, D. We're gonna kick it old school here. Use pens and paper. Pretty exciting. And keep these for the future. Come on, what are you doing? An A, B, C, D. You should A, B, C, D. And I'm gonna go through the quiz or selections of questions from the quiz, and you guys are gonna hold up your answers. And that way I can engage if people are, but if everyone has the same answer, then I feel good. If there's a lot of different answers, I feel bad. And then you guys also feel like you know what's going on. Okay. I see some people are ready. Who watched that football game last night? I'm telling you, if you want to see crazy, come to my neighborhood. The minute that game was over, screaming in the street, screaming, fireworks in front of my house, for real, for real. I don't know, maybe you guys had that up here in Towson. Fireworks?
Yeah. Yep.
Crazy. And on Facebook, all my friends with kids are all like, you better not wake up my kids. My kids don't wake up. Okay. Time is about to run out. Okay. The following is true of sperm cells. Sperm cells are diploid, haploid, polyploid, haploid. What do you guys think the answer was here? Okay, good. I see some D's and some B's. Okay. Well, I am really glad there's some B's up here. Okay, so haploid, you guys understand that means one copy. So that means individual chromosomes. So B and D are confusing in a way. But if you pay attention, you'll see that B says containing 23 pairs of homologous chromosomes. So by them being paired with homologous chromosomes, sperm cells are that last phase of meiosis, where there's no more splitting to be done, so you've got one copy. So I have that there. So you can see here, that's where your sperm cells are. So there's no more homologous anything, there's no sister chromatids, there's nothing. You got one copy, and that's because they're going to have to meet up with an egg. And then you don't want to have, you need one copy from mom, one copy from dad, right? Okay. Keep it easy here. Okay, karyotyping, a type of chromosome analysis, must be performed during what phase of the cell cycle? Okay, good. Everyone's saying B. Great. Why don't you wanna do it in interphase? They're not condensed chromosomes. Absolutely. Okay. That's good. That's a good thing to understand. Any questions? Good. Okay, chromatin is made of lots of D's. I see A's.
One website that said
There's a website that said A?
yeah.
DNA, RNA, and protein. What? Okay. Chromatin is your DNA all wound up with your histone. That's pretty much what chromatin is. Where would the RNA fit into that? Okay, send me the link.
Oh yeah. I didn't use that.
I know, but so this is the thing about this class. It's so important to be resourceful because sometimes you can't find the answer in the book. Sometimes you call your friend and your friend doesn't know either. Sometimes you do need to Google it, and that's okay, and that's important because it's called being resourceful in life. But also you need to really think about that answer and say, okay, who wrote this? Because people have these biology blogs where they write crazy things. You just never know. Wikipedia, I had to fix the Wikipedia entry for HIV because it was wrong and outdated. So just be very careful. Personally fixed it. They said there was no known function for VIF, and I was like, dude, it's like 2005. Anyways, it's okay. Okay, so the answer was DNA and protein. Okay. The purpose of histones is to C's, lots of C's, some D's, C's and D's. Okay. Replicate DNA, repair damaged DNA, condense DNA so that it can fit into the nucleus. Definitely C. And I think I know why I see some D's. Because you're probably thinking about how histones need to be removed to replicate DNA, or how there are histone variants that are involved with signaling the need to repair DNA, but they do not directly do those things. Okay. Nope. Okay. Wait a minute, chromatin? Oh, nucleosomes. That was the same picture. Nucleosomes are made up of D's and D's and D's and D's. Very good. I hope that's right. Histones and DNA. Right. That was an obvious one. Okay. Histone tails play a direct role in well, there's definite interaction with adjacent nucleosomes because that's how the condensation occurs. And then they're really important in chromatin structure and function, right? So that would be D. Good. Okay. Histones are more than just DNA packaging machines. Histone variants act in diverse capacities. See all, select all that apply. Okay, so which one does not? Which one does not? E. Right. Histones do not assemble ribosomes. That was like I was searching for something.
Could you explain that X inactivation?
Oh, okay. We will go into that as we talk about it. X inactivation. So, how many X chromosomes do you have? 2. Okay. And so, sorry guys, you're not part of this discussion. So, did you ever wonder why, like, if there's one of every other chromosome, but we have 2 X's, we have twice as much dose of genes coming from the X chromosomes because we've got 2. No, we got to turn one off. So that's what X inactivation refers to, and there's a histone variant that is present on the inactive X chromosome. Okay, good question. Any questions about this one?
I was having trouble finding information about DNA image sensor.
That is H2AX. Was it not anywhere?
Oh, I mean, I looked through the book, but
Okay.
I missed it.
Okay, I'll look it up. So sometimes, here's a little tip with a quiz taking. So it's good to have your book in front of you, but it's also good to have the NCBI site up so you can search it.
Well, I have a PDF copy, so I searched
Good.
the whole thing for iWords.
Okay, so I'll have to look it up. Shoot me an email so I can see if it's okay. Yes.
Would you consider histones and variant histones together?
You can. They have the same function. Histones and variant histones all form the histone, but in different. So you've got histones that are made up of different subunits, right? And these variant histones are just different subunits, so they swap out. So H2A is one, but there's H2AX that, in cases of DNA damage, will replace H2A in forming the histone. Does that make sense? So we'll go. This is just a quiz, so we will learn this in more detail. Let me see if I can. Let's go back here just for a second. See how it's an octamer, and each subunit of the histone. So this is a full histone, right? Assembled. but then these are the different subunits. So here's H2A, this is a normal subunit that makes up a full histone. And then with DNA damage, the H2A is going to come off, and you're going to actually have to be replaced with H2A acts, which looks different, and acts to recruit DNA damage repair proteins to that damaged section of DNA. So it's like it's a histone. It's just that there's a variant in a subunit. Okay. Okay. A gene that is being transcribed is most likely to be found. Now, this required you to actually do some thinking into what DNase is and what DNA sensitivity or resistance means. So if it's being transcribed, it should be in open or closed chromatin. Right, so in euchromatin. And if you have open chromatin, would you be sensitive or resistant? Sensitive. Right, so everyone's got D. Very good, yes.
What does DNase want to do?
DNA cleaves DNA, so it degrades DNA. So the way to think about it is if we go back to this picture here, you see how you have your DNA wrapped around your histone? So if you've got this tightly packed heterochromatin, then you've got your DNA completely obscured and wrapped around histones, and you can't get your DNA in there. It just can't physically fit in there to cleave the DNA. So it's resistant to DNA.
So it cleaves the DNA that is being transcribed?
Right. And it's not that it's being transcribed, it's that the DNA is going to be open in an area of transcription.
So
Okay.
heterochromatin isn't open.
Great. Right. Heterochromatin is compact, euchromatin is more open and likely to be areas of active transcription. Yes?
Is this cleaving damage to DNA?
This is experimental. It's not something that happens in the cell. So let's say, so it's more like if I gave you a tool and I said, hey, tell me if if I gave you a sample of DNA and said, tell me if this is heterochromatin or euchromatin, if it was sensitive to DNase, then it would likely be euchromatin. Okay, you guys can go.